A technique that amplifies DNA with high specificity might sound like PCR. Today, however, we have a look at another DNA amplification method, which is termed LAMP, loop-mediated isothermal amplification. This technique has some advantages over polymerase chain reaction and is used a lot in diagnostics. Before explaining how LAMP amplifies DNA, make sure to subscribe to this channel for more scientific content. PCR definitely is the most famous genome amplification technique. Why should one use LAMP instead of PCR? LAMP is much faster in amplifying a DNA sequence. Apart from that, the second advantage is given by the name. Isothermal means the whole reaction can be performed at a single constant temperature. PCR, in contrast, is based on cycles with different temperatures. This requires expensive machines. LAMP requires 4 to 6 primers. 4 are essential, and it is recommended to use 2 additional loop primers to facilitate product amplification further. But how does LAMP work? This is the double-stranded DNA that also contains the target sequence. The regions upstream and downstream of our amplicon are labeled to illustrate primer binding. The reaction starts with strand invasion, and the forward inner primer F2, complementary to the F2C region, initiates synthesis executed by a special polymerase. You may have noticed the 5' overhang of that primer. It has the reverse complementary sequence to that of the F1 region. This will be important later. Now, the forward outer primer binds to the F3C region with the only purpose to dissociate the gray graded DNA, which is now single-stranded. What has happened for the forward sequence will now also occur with the backward inner primer. B2 hybridizes with the B2C part and initiates synthesis of the complementary strand. Again, you may have observed the 5' overhang of the primer is the reverse complementary sequence of B1. Good. Now the outer backward primer hybridizes to the DNA strand, and as a consequence of this, the upper DNA sequence will dissociate. This will be our first product. Now, the reverse complementary sequence F1 hybridizes with the F1C part, forming a loop structure. The same happens with the backward sequence B1 and B1C. The product takes a so-called dumbbell structure. From here on, all primers can bind to generate more product. Also, loop primers enhance product amplification. The LAMP reaction yields many different products. In the end, we need to determine whether a sample is positive or negative, meaning if it is containing or does not contain a specific DNA sequence. There are multiple ways to detect LAMP products. One would be to simply use DNA binding dyes. As in qPCR, the products can also be quantified. With that, I thank you for watching. If it was helpful, please like this video. Here is a video that briefly explains the mechanism of the polymerase chain reaction. Make sure to subscribe for more scientific videos. See you next time.